The overall goal of the PIP-On-A-Chip assay is to assess protein membrane interactions in a quantitative label-free manner.
Protein membrane interactions are at the heart of so many processes of the cell and its pathogens, but techniques to study these interactions are few.
The advantages of this technique are low sample volume, no ligand or receptor labeling requirements, combined with the ability to test membrane interactions in a physiologically relevant manner.
Many therapeutic targets of viruses are membrane proteins. Studies of these target proteins are often performed in solution using detergents. Our technique provides a more biologically relevant alternative.
While you will see that this assay is capable of monitoring the interactions of proteins with membranes, it's actually quite a versatile assay and can be used to monitor ion membrane, small molecule membrane, and even peptide membrane interactions.
To begin, mix polydimethylsiloxane, or PDMS prepolymer, and the curing agent at a 10:1 ratio in a large plastic weigh boat. Degas the mixture in a vacuum for 1 hour with a vacuum strength at 500 torr or less. Place the silicon master, which contains multiple replicates of the same SU8 micropattern, in a large plastic weigh boat and pour in the degassed PDMS. Then, cure it in a dry oven at 60 degrees Celsius overnight. The next day, gently peel off the PDMS from the silicon master using one's hands. Mark the boundaries of each micropattern in rectangles using a surgical scalpel and a ruler. Then, cut the PDMS into blocks.